So we've investigated the frogs using a variety of different things. We've looked at peptides to investigate, and this is the red-eyed tree frog you've just seen, but it comes from a variety of different areas. I said before, Mexico to Panama, different colours in different areas. And different things affect the colours. So different things that might, perhaps an insect that lives in Mexico doesn't live in Panama, so that's going to have an effect on the frog's colour. But also the peptides in its skin that keeps it alive. It's almost like the poisons. Each one's got its own peptide profile, if you will. And by looking at that, we can actually use it for taxonomy, so we can see which ones are more related to others. We can kind of investigate them. And I'm really quite interested in speciation and how they evolve to be separate. So over the last few years, this was quite a few years ago, about four or 5 years ago, when I went to Thailand, we started to develop ways of looking at the DNA and collecting DNA without hurting the frogs. Up until then, the only way to do it was to actually chop the little toe pads off. It sounds pretty horrible, and it is, because most of them die afterwards. But that was the standard way to collect DNA from the frogs. So we trialled a way of actually swabbing them from the mouths, and it actually worked pretty well, so we kind of developed that. But we were looking at different populations. These are both adult males from different populations. You can see the difference in the size. And the reason why it's important to kind of differentiate between different species, well, between different populations, even of the same species, is critical if you're trying to conserve them. Okay, this is a frog called the lemur leaf frog. Okay, some of you might be familiar with it. It's critically endangered. It's probably one of the most endangered of all the leaf frogs that we've got. We started the program for this one in 2001. I was given special permission to collect a few eggs from the Costa Rican authorities. We bred 400 of them at the museum. They've been sent out to different zoos worldwide, including Atlanta Botanical Gardens, Vancouver Aquarium, and in this country, Bristol Zoo have got some. But it's a critically endangered frog. It's only known from about one pond in Costa Rica. It goes brown at night. It's actually bright green during the day. And it changes colour. This is a little guy here. This is a female. She's quite big, actually. There we go. See it there. So she's absolutely bright green during the day. As soon as it goes dark, as soon as the lights go out, she changes. She goes brown, chocolate brown, and even her eyes change colour. Really cool little thing. Critically endangered, only known from the one pond in Costa Rica. They're called lemur leaf frogs because they kind of walk around rather than jump. A bit like a lemur from Madagascar. Okay. But that particular frog, the lemur, is known from one pond in Costa Rica, but a few different sites in Panama. When we did some DNA work looking at that, we actually found that there were two very, very separate populations. They're actually the same distance between them as two other completely different species. So even though they're critically endangered, they're probably even more critically endangered if you think of them as two separate species, and the fact that that one species is only known from that one site. So that's the reason why we started doing some of this DNA work, to actually see where we could focus our conservation efforts. And here we have this is a little baby lemur, tiny little baby one. It's about 2 weeks old. I'm not going to wake him up.